Hello and welcome to Ask Your Academic. Today's session will cover the MSc Biotechnology, the MSc Biotechnology and Management, MSc Chemical Biology and the MSc Stem Cell Engineering for Regenerative Medicine. We're joined by a number of our programme leads today, and we will put the questions that have been submitted towards them. If you have any questions throughout today's session, you can use the chat box and we'll do our best to get to those. If after the session you have any further questions, you can send an email to the programme administrator and they'll do their best to redirect your query so you get your answer. We are recording the session today and that link will be made available to you afterwards, probably once all our sessions are complete. So we do only have a very short session, so if it's okay with everybody, we'll get started on the questions. Our first question is, what kind of career support is being provided to the students after completing the programme? Cheryl?
Shall I take that one? Hi there, everybody. So my name is Professor Cheryl Warhead. I am the Dean of Postgraduate Teaching for the College of Medical, Veterinary and Life Sciences. I'm also the Programme Director for the Biotechnology and Management Programme and teach on all of the programmes that we're discussing here today. So hopefully myself and my colleagues will be able to answer the majority of these questions. So in terms of career support, we definitely provide careers service days for links with industry and the opportunity to find employment after you graduate the university throughout your time here at the university. We also offer support with CV writing and interview technique and the ability to sort of write letters to companies if you're looking for a job afterwards. In addition, we also provide some PGT to PGR conversion events where you can investigate if you wish to do a PhD after you finish your master's degree, and that will give you the opportunity to hear about specific projects happening within our institute school, within the College of Medical, Veterinary and Life Sciences throughout your degree. Just finally, in addition, I would say that all the programs that we're talking about today are accredited by the Royal Society of Biology. And on there, you'll gain a year's membership to the Royal Society of Biology as part of your MSc degree. And from there, there's obviously ability to network and look at other avenues and support services for careers throughout your study. Okay. Thanks, Naomi.
Thanks. Is there any practical work on using the induced pluripotent stem cells and 3D printing techniques? Catherine, is that one for yourself?
Yes, that's right, yeah. So hi there, I'm Dr. Catherine Berry. So I'm the programme lead on stem cells for regenerative medicine, stem cell engineering. So we work with several types of stem cells in our labs, mainly adult stem cells, mesenchymal stem cells and hemopoietic, the blood stem cells. We also work with cancer stem cells and leukemic stem cells. There's a little bit of work that is done on human-induced pluripotent stem cells, mainly in deriving them towards cardiac myocytes with one of the colleagues that we work with. So there is potential to do a little bit of work in that area. In terms of 3D printing, that is something we do quite routinely in the lab. So we 3D print various materials in order to get our cells to interact with them. And we also do 3D printing and bioprinting of bioinks. So that is using various different hydrogels and bioinks with cells combined in order to either specifically print into a very small area or to print specific patterns. So that is potentially something. Now, these types of work tend to get done more in semester 3 in a research project. So all students have a research project within our labs whereby they can select a project that suits their area. So 3D printing is something you're interested in. Typically, we do have a few projects using that technique. Okay.
Thank you. What are the possible laboratory facilities for stem cell research at the university?
So almost everybody who supervises a project is now relocated in the Advanced Research Centre or the ARC building in Glasgow. And that's where we do most of our stem cell research now. Within that, we have the usual tissue culture facilities, and we also have a lot of microscopy, including we work a lot with 3D culture and hydrogel. So we have atomic force microscopy, biorheology, we have confocal, we have fluorescence, all these standard techniques that we use to be able to visualize our cells in 3D. So most of the work will get done in these labs here. Yeah.
Great, thank you. If we are interested in the research work of a particular professor, is it encouraged to contribute to their work?
Well, it depends. If the professor is involved in CME, which is the centre that I am in MVLS. So we are the Centre for the Cellular Microenvironment, or CME, and all the staff members or project investigators, PIs in CME, offer projects to students, at least 2 or 3 each, if not 4. So if there's a professor you're particularly interested in who perhaps works in an area you want to work in, and they are within CME, then they will be offering a project and you can work with them and therefore contribute to their work in that way. That's something you could probably email me directly about if there's somebody you're particularly interested in. I can let you know whether or not they are involved in this programme, so you know ahead of time.
Great, thank you. Does the Biotechnology and Management programme intend to develop bioinformatic or programming skills?
Shall I do that one? So biotechnology and management isn't a strong bioinformatics type program. We have other programs which are using a lot more bioinformatics than the biotechnology and management program. You are, however, offered a course in Introduction to Bioinformatics, which you can take if you choose to. But the majority of the program is split between our core biotechnology skills and the management courses, which consist of project management, change management, HR management, marketing, creativity and innovation, and strategic change. So those are the main marketing courses that we will take as part of the program, but there is an opportunity to take an introductory course to bioinformatics. Thank you.
Perfect, thanks. Are there any extracurricular workshops that teach more about cellular, molecular, or immunological techniques such as Western blot or immunoassay? I think I said that right, hopefully.
I think you did. You did a good job. It's not an easy question. We do not run extracurricular workshops on laboratory skills. As part of any of these particular programs that we're talking about today, you will get a significant amount of time doing laboratory skills as part of the program. PGT degrees are quite intense, just to give you that update. There is not a lot of room for extracurricular study around the amount of work that you'll be doing. It's a 1-year, quite intense, hardworking program. So we teach you everything that we can within that 12-month period. The only extracurricular things that you'll find are sort of in skills development, which you can do optionally on top of your program.
Thank you. What is the main differences between the biotechnology projects and the dissertation project?
Sean, do you want to take that one?
I can try and take that one. So I should introduce myself. I'm Dr. Sean Collins. I'm program lead for the MSc in biotechnology. So the course starts off with 120 credits of taught courses, and then everybody goes on and does a 60-credit summer project of some sort. So those projects, a large number of them, take place in laboratories of researchers within the university. And so those will be the lab-based ones. You'll join a research group, take part in some of the research that's taking place in the lab, contribute to that research, and at the end of it, write up a report of the research you've done. The dissertation projects are more, they're research-based, but your research will be the scientific literature. So you'll be associated with a laboratory or a researcher within the university, but they will just be directing you to develop a question, directing you towards literature, scientific papers to read. You'll read those papers and try to answer some sort of scientific question. And at the end of it, again, you write up a report summarizing your findings. Is that fair enough?
Thank you. Sean, another one for yourself, I think here. Could you give us a brief explanation of the course structure, the syllabus and exam pattern? And do we need to study some programming language?
That's 3 questions in one. So Let's find this so I can remember. I've already forgotten them now. So the course structure. So I said a little bit about that in my last answer. So there's 120 credits of taught courses. So that starts with an introductory course to molecular research skills, where you learn the basic techniques of molecular biology. You then go on and do industrial environmental microbiology, where there's some lectures, and then a lab practical where you look at antibiotic production by bacteria. There's a course on biotechnology applications, one on protein expression that Cheryl teaches, where you learn how to express proteins and how to run protein gels. And maybe even, is there a Western blot in that one, coming on from a previous question? Then there's a choice of 2 courses, one on genome editing, one on technology transfer. And then there's a choice of multiple courses, one of which is the bioinformatics course that Cheryl previously mentioned. Other ones are bioimaging, omics, or a plant biotechnology course. And then we finish with a course I teach, which is synthetic biology, how to reprogram DNA by changing the DNA. And then you go on and do the 60 credit project that I talked about in my last answer. There was more to this question, wasn't there?
Yes, so the exam pattern.
So assessment takes place via a variety of methods. So there's assessment associated with each of those 10 or 20 credit courses in the taught part of the course. And those tend to be things like lab reports, writing essays, doing spoken presentations or poster presentations, and some exams possibly. So, but those again would be essays under exam conditions. And then of course for the project, there is the main part of the assessment for the project is a written report at the end of the project.
And just that last little bit.
Programming languages. So again, there's no formal requirement for writing computer, for doing any computer programming in the course. There'll be a little bit of coding associated with the bioinformatics course. That's one of the optional courses. If you're a keen programmer, go ahead and teach yourself a bit of Python or something like that. It's always useful before you come, but there's absolutely no requirement for any programming.
Thank you. And Cheryl, one for yourself. Could we have more information about the Industrial Networking Symposium and Industrial Networking Sessions?
So throughout the year, we have some external speakers which will come and speak to all the students from industry. Everybody would have the opportunity to attend those sessions and then to meet in small groups with speakers after those sessions would be. Those are not part of your curriculum. That's an additional voluntary participation that you can do if you wish to. So those are on top of what your actual classes are. We also have a lot of external speakers come to do the technology transfer and bioscience commercialization course, which runs at the beginning of semester 2. So there'll be additional industry talks within that particular course.
Thank you. And how many of my classmates will take classes together?
Shall I start with that one? So on all of the different programs that we're talking about today, there is different numbers of enrolled students that will be taking those particular courses. What you'll find is that in some aspects, those courses are then taken with others. So for example, at the beginning, all of these actually 3 of these 4 courses will take classes together, and there'll be quite a large class, and you'll be in lab groups between about 60 and 70 students in a lab together. Generally, the biotechnology program is the largest, so there will be probably about 80 to 90 students on the biotechnology program. And in some cases, you'll take classes in one large group, and in some cases, you'll be split down into smaller groups. The biotechnology management program and the stem cell engineering program tend to run between about 20 and 30, and the chemical biology program is around 10 to 15 students on that program.
Thank you. Anybody else want to add anything to that one? Are we all good?
Fine, Cheryl's covered ours really. There's usually between 20 to 30 students on the stem cell engineering program, and there's several courses where you will overlap. There's 2 engineering courses, for example, where you will overlap with other students in engineering, and there's a couple of courses where you will be able to do options, so you will split yourselves essentially. There's some core courses and some optional courses, and the optional courses you will split and choose what you're interested in semester 2. So that way you'll be working with people in your class and other people from other cohorts.
And that goes for all of the programs that we're doing optional courses and mixing with other cohorts in semester 2.
Thank you. Brian, one for yourself. Is it possible to specialise in plant biotechnology?
I think Sean should take that one maybe.
Oh, sorry.
I'll take that one. So that's a biotechnology question, I believe. So there's no specialisms as such in the MSc in biotechnology, but there is one of the optional courses we have is the plant biotechnology option. So you can do that course. So students really seem to like that course. The ones with interest in crops and plant sciences, they really like that. It's a great course. And then of course, when you come to do your project, there are some projects that are available in the subject area of plants and crops. Anything to add to that, Cheryl?
Nope, I don't think so.
Thank you. And what is the main building the MSc Biotechnology and Management will be based at?
So that program is based purely on the main campus of the university, as all of these programs are. The management side will be based out of the Adam Smith Business School, which is actually in the main building of the university on the Gilmer Hill campus. The biotechnology side will be based between some different buildings on campus, the Boyd Orr building, the Joseph Black building, the Bauer building. But all of those buildings are within a 100-meter radius of the center of the campus.
Thank you. Are the programs expected to be taught in person, online, or with a hybrid learning?
Shall I take that one?
Yeah, you can, Joe.
You can go to it, Catherine, if you want. Yes, we are expecting to teach everything in person by September. There may be some information or parts of courses, particular sessions, which are delivered online for a particular reason. If it's utilizing a particular software or something, that there may be some particular sessions which will be delivered online, but the majority of the teaching will now be on campus in person as of September.
Thank you. Is it possible to do research in any industry? It's not got a program specific, so if there's anybody that would like.
No, for our one, we did try this a couple of years ago, and we got 2 companies involved, and neither of them came through in the end. So we believed it's actually safer to have the students on campus under our guidance in the final semester project. Yeah.
Yeah, if I would just add to that, even though a third of the year is dedicated to the project, it is not really long enough to become embedded in an industry placement and to experience a good student experience during that time period. So we tend to keep majority of the projects in-house, and so people are able to get up and running a lot quicker and to learn a lot more and appreciate the experience of the project a lot better than they would do if we sent them to external industries. That doesn't mean that the research work you will be doing may not have an industry link. We may have some academics who are working with different industries to investigate a research question, and that might be possible, but we will, as Catherine says, the majority of the projects will be based on the site of the university.
And we've got excuse me, a question that's come through our Q&A box, and it's for Sean. What references or books would you suggest for us to get a gist of synthetic biology before the session begins?
I was thinking I was going to suggest maybe some more general books for biotechnology, just like an introduction to biotechnology textbook, like say Godby, Introduction to Biotechnology. So that might be sort of good, more general background reading before you start, because for the synthetic biology course, you do need to sort of understand basic genetics, processes that turn a DNA into a protein. So make sure that you're really happy with understanding how transcription and translation take place in cells, so that DNA can be turned into proteins that do things. The other interesting thing to find out about is the actual iGEM competition. So that's IGEM. So this is an international competition that we sort of loosely base the synthetic biology course around. So if you find out what students have been doing on the iGEM website, I can post you a link to that, or I can get Naomi to post a link to that maybe. Have a look at that, and have a look at projects that have been done in the area of synthetic biology by student teams all over the world.
Thank you. And I suppose for yourself, Cheryl and Catherine, is there anything that you would suggest for anybody starting? Is there any proposed reading or any suggestions for that for getting started?
Was that what was it in terms of reading material or in
Reading
terms?
material? Yeah.
Yeah. So what I usually tell students is to contact me, because I've got a few start-off papers. It's more, I'd suggest a couple of review articles rather than a general textbook would get you in the right field for the right area for doing the types of work that we will do in projects and things like that, and you will get taught about. So I tend to tell students to contact me directly, and I can send them something on that I've got prepared.
So in terms of biotechnology and management, the same would apply as what Sean says for that particular program. It is very biotechnology based. The management side of it, you'll get the information at the beginning of term. The Adam Smith Business School will run that side of the degree, and they will provide you with appropriate management literature to get you up to speed when you start.
Thank you. And we do have a couple of other questions in there, but they are more to do with the admissions. And we'll not be able to address those questions on this webinar just because we are so short for time. But I have posted some helpful links into the chat box. So for anything admissions related, you can pick up with them and they'll be able to advise you further. I'll just have a look to see if we've got anything else coming through.
There's a question about the stem cell engineering program structure. I don't know, Catherine, if you want to maybe.
Yeah, both of these questions have come through on the chat box. I actually answered them on there, but I don't know if that's visible to everybody or not. So I've answered them for Mariam's question about the structure and the syllabus of the program, saying that, yeah, I have documents for that, and to explain if I've given my email address to them on the chat box, saying, please email me directly and I'll send you that. And I've also suggested topics in the chat box that we do work on in research projects, but we do not cover neurological and neurodegenerative diseases. We do cover a bit of peripheral nerve repair, but we don't cover actual disease profiles. So it's more adult stem cells with MSCs, HSCs, and leukemic stem cells, and looking at stem cell interaction with material surfaces in 3D, in hydrogels, things like that. So it's not actually a disease profile for neurodegeneration. And in terms of preparing for the cell engineering program at the start of the study, like I just mentioned then, again, email me directly and I will give you some material that you can have a look at. Okay.
That's great. Thank you very much, Catherine. Well, that's 12 o'clock. That's the end of our session, I'm afraid. We hope you've found everything that you've all the answers to the questions that you've put towards us. If, like I said, you've anything further, you can contact the administrators of the programs and they'll do their best to get back to you with the answers. So thanks very much everybody for all your time. We hope you take care and we hope to see you all very, very soon. Thanks very much. Bye-bye.
Thank you. Bye.